Thank you, Brett. I'll reiterate what you heard from everybody, which is it's a pleasure to be here and to be on the panel. What Luminex does and what companies in our industry do is somewhat unique in that it requires expertise in a number of different disciplines. All the different engineering disciplines are required: mechanical engineering, electrical engineering, software, optical engineers, et cetera. In addition to that, all the various biological disciplines, immunology, genome expertise, et cetera, are also required in order to deliver to the marketplace the necessary products and solutions. And that's an important theme that you'll hear through my remarks. So if I could have the first slide, please. I did. There we go. Okay. So you'll hear through my remarks the importance of this cross-disciplinary approach. Let's start with the fundamental understanding that is continuing to develop around protein research. In the state of Texas, of course, a Nobel Prize that was awarded to Dr. Gilman for his research around proteins and cellular activity. And researchers around the world continue to pursue that level of understanding, how cells interact and how they signal with each other. Among the scientific innovations, however, that's been crucial to continuing to make progress in that understanding is the fundamental detection technologies. That's the engineering and the scientific expertise I just mentioned, to be able to actually do the measurement, do the detection, and come up with the fundamental data to arrive at appropriate scientific conclusions. And more recently, in addition to proteins, we're all aware of all the progress that's been made on the genomic side with our understanding of DNA. Now, genomic research is very well known and is very well publicized. We're all aware of the Human Genome Project. But what I would ask you to think about when you consider the Human Genome Project and the mapping of the human genome is imagine you finding yourself in the middle of New York City and you need a phone number. You go to the phone booth, assuming there is one. You open the phone book and you find millions of phone numbers. What you don't find is any name or any address. You just have phone numbers. That phone book, without names and addresses, is the equivalent of the Human Genome Project. In other words, the only way that information becomes relevant is if we understand what a specific genetic sequence or genetic mutation means in a clinical sense. And so the understanding and the linkage of DNA and proteins is crucial in that effort to understand how DNA sequences and DNA mutations manifest themselves in a clinical way. There are millions of cases that are documented where two humans with identical genetic mutations have very different phenotypes, that is, very different presentation in a clinical sense. So the interaction between DNA and proteins and the clinical presentation is extremely important to understand. So the Human Genome Project is now being followed up in a number of ways. One of those is the Protein Atlas Project, which is a collaboration in Stockholm between the Royal Institute and Uppsala University, and they have recently announced the fact that they have completed 50% of the categorization of the known human proteins. That's a very important concept to consider as I go through my remarks. At the fundamental backbone of this, however, is the innovations are possible as a result of the detection technologies, which in turn, as we heard earlier today from Dr. Mendelsohn, are making progress, allowing us to make progress in this concept of what is called personalized medicine. And that's what the example that I'll use. So as I mentioned, genetic signatures can vary from person to person. How those genetic signatures are manifested by an individual also vary from person to person. And how those people respond to those different therapeutic regimens also vary. It's true in small molecules like pharmaceuticals, large molecules from biotech companies such as monoclonal antibodies, and all the way to a therapeutic regimen that may be an organ transplant where there's testing that needs to be done in order to determine whether an organ recipient and an organ donor are compatible. The promise of personalized medicine, that is, using that that basic information that's different from person to person to tailor an appropriate therapeutic response is much more than what you may have heard, which is the right drug with the right dose to the right patient at the right time. An example of that, which I have on the slide, is a very well-known example, the cancer drug tamoxifen, which is metabolized in the body. It's what's called a prodrug, which basically means it turns into its active ingredient as a result of metabolism. There's a genetic mutation that determines from person to person how well that patient can metabolize that particular compound. It's called the 2D6 gene, and that's just an example where an advantage to the patient and to the physician and the healthcare system is to make sure that the patient will actually benefit from the therapeutic regimen that's being introduced. So if you agree that it's important to understand not just what's happening at the genetic level, but also what's happening at the cellular level and with proteins, then there's a significant opportunity to be able to test both DNA and proteins simultaneously. However, a significant issue is that all known methods of amplifying and detecting DNA effectively denature all the proteins that are present in that particular sample. So the solution that's being pursued by Luminex in particular, but also by others, is to find ways to converge existing technologies to increase the opportunity for scientists and researchers to identify both proteins and DNA in the same sample and therefore get more relevant, more clinically relevant information. Many times that requires multiplexing, which means detecting multiple targets in an individual sample at the same time. What we're talking about here, of course, is expanding that to not just DNA targets or protein targets, but both DNA and protein targets in the same sample at the same time. Some of those technologies are the application of known technologies such as electronic circuitry and electrowetting. The implications, of course, are that by ensuring that the patient receives the right therapeutic intervention at the right time and that they will benefit from that therapy will allow us to have a significant effect not just for that individual patient, but to ensure the healthcare dollars we're spending are being more effectively spent. Now, so what are the barriers to that? First of all, there are significant scientific challenges associated with that particular effort as we continue to increase our level of understanding of the molecular mechanisms, what's happening at the DNA level and what's happening at the cellular level, doing the work to connect the phone numbers to the names and addresses, that is, understanding what markers are associated with different disease states. There are significant economic challenges to ensure that the alignment of incentives are appropriate among the various stakeholders. I would remind you as an example that in our industry, that is the medical device diagnostics, pharmaceutical, and biotech industry, virtually all the innovation takes place in smaller companies. We all see news reports of how smaller companies are acquired by the big multinational companies, and that is just a fact. What is an interesting thing to consider, I think, is the recent Supreme Court decision that lifted the restriction on corporate contributions to political campaigns, and the fact that a lot of those large multinationals have a significant investment in the maintenance of the status quo, because you have these large companies who have built their entire enterprises around the existing healthcare delivery system, and it's important to consider that investing in that status quo and maintaining that status quo is of paramount importance to a lot of those companies. So there's significant economic challenges there, the operational challenges to ensure that the reimbursement systems are aligned, and of course that physicians who control the ultimate disposition of all healthcare are appropriately educated. We have a saying in our company which is the most expensive and the most powerful medical device known to man is the pen in the physician's pocket, because it's that pen that determines what healthcare is delivered, and the physician therefore has to be educated as to what these latest innovations are. There are also legal challenges. There's currently cases in the court system around the patentability of naturally occurring substances, including biomarkers. I listed a couple of those on the slide, and the trend is toward the fact that these biomarkers will be patentable. That's an important factor to consider, because if they are patentable, you create the incentives for some of the innovation to take place. There's certainly an argument to be made on the other side of the coin about about these biomarkers not being patentable, but there's a price to be paid for that because of the lack of a financial incentive. And then, of course, the regulatory challenges with, in this country with US FDA. As you may know, there's a lot of changes underfoot or underway at FDA, including the revising of the review process, particularly for medical devices. That is likely to be more time-consuming with more expensive clinical trials and so on. And that's not really a judgmental statement, it's just a fact. And so that will also affect the way in which these innovations are deployed. So as we look toward the future, personalized medicine and its cousins all have great potential, but there are also significant challenges associated with them. Obviously, an ongoing commitment to research and development. In our industry, the rule of thumb is companies typically spend between 15 and 20% of revenue in R&D. My company and many others are within that range, and that's a significant commitment to innovation, of course, as we use these enabling technologies to solve the ongoing technical problems. I mentioned the tough public policy choices that are required to ensure that we get everybody aligned appropriately. The government agency harmonization in our industry, the reimbursement system, the CMS, FDA, et cetera, to ensure we don't have products that can save lives and save money that are not reimbursed, and we don't have products ready for reimbursement that are not yet approved. And that will require a significant amount of leadership from the FDA, other regulatory bodies, as well as organizations like CMS. And then lastly, as I mentioned a few minutes ago, the importance of physician education around these new concepts is extremely important, and will require a commitment by the educational system to ensure that with these innovations underway, that physicians are appropriately educated when they're in medical school, if not before, about things like DNA and the interaction of DNA and proteins, and to make some significant progress there will be extremely important. So those are the end of my remarks, and I'll turn it over to the next speaker. Thank you.